Hello judges, my project aims to tackle hydrocephalus, an incurable neurological disorder characterized by abnormal expansion of brain ventricles due to impaired cerebrospinal fluid circulation. Hydrocephalus affects nearly 1 out of every 500 newborns, making it as common as Down syndrome. In the acute stage, dilated ventricles and increased intracranial pressure causes symptoms such as headaches and nausea. In the chronic stage, fluid dynamics normalize, resulting in dementia-like cognitive impairment in memory, information processing, and other critical functions. The principal neuron of the brain's hippocampus is the pyramidal neuron. As the most abundant cell type in mammalian cortical structures, it is believed to play a pivotal role in higher-level cognitive functions. Both dendrites and their spines, located along pyramidal neurons, are important structures with crucial roles in brain connectivity. Dendrites serve as the main postsynaptic target of excitatory synaptic inputs, while dendritic spine dysfunction has been implicated to be a common substrate among pathologies of many neurological disorders. Although hydrocephalus is known to induce axonal degeneration and demyelination of white matter, its effect on neuronal structure remains largely unknown. Furthermore, changes of the hippocampus in any hydrocephalus model have rarely ever been examined. While alterations of hippocampal pyramidal neuronal dendrites are studied in disorders such as Alzheimer's and trauma, little is known about the effects of hydrocephalus on the dendrites of pyramidal neurons. For these reasons, a more comprehensive evaluation of the effects of hydrocephalus is needed. The first objective of my experiment was then to evaluate the dendritic structural alterations that occur in the hippocampus during hydrocephalus. The two factors I tested for were dendritic length and spine density. I chose to restrict my investigation to the CA1 hippocampal region, as it is the most involved in spatial learning and memory. Additionally, I examined only secondary and tertiary dendrites, as they are homogeneous in regard to dendritic morphology. My other two objectives were to identify responsible mechanisms for the deficits associated with this condition and to further our understanding of its pathophysiology. The methodology of this project can be split up into 4 distinct sections, the first of which, an experimental hydrocephalic mouse group, was generated utilizing a Krelok system, while littermate wild-type mice served as controls. It should be noted that only male mice were used to avoid any fluctuations in spine density that occur during the female estrous cycle. Next, mice were sacrificed on week 5 and perfused. The dissected brains were then equilibrated in sucrose solution and embedded in freezing compound until further use. Later, brain samples underwent the Golgi staining process, during which they were immersed in silver nitrate impregnation solution, allowing sparse labeling for data analysis. Tissues were then dehydrated using an alcohol series, and sections of 50 micrometers were then obtained. Lastly, the Golgi fixed brain sections were converted into image stacks using the Leica application suite, and then dendritic lengths and spine densities were calculated using ImageJ analysis software. The initial qualitative assessment showed that mice with hydrocephalus revealed 3 distinct features: smaller arborization of dendrites, fewer apical dendrites, and fewer dendritic spines. These observations were supported by measurements from ImageJ, revealing that the mean dendritic lengths and spine densities were significantly reduced in the hydrocephalic groups compared to the length and spine densities in their age-matched controls. After conducting a 2-sample t-test, I found that the reduction of the mean spine density in hydrocephalic mice was statistically significant with a p-value less than 0.05. Findings from this study suggest impaired excitatory connectivity, which could underlie the learning and memory deficits associated with hydrocephalus. With no established methods for automated 3D neuronal analysis, taking into consideration all spine protrusions posed a challenge. Much of the data collection instead relied on manual computation, which was time-consuming and subjected to bias. In the future, I want to conduct my experiment under different chronicities of hydrocephalus. I hope to introduce more subjects and study regions, and to also implement my findings into clinical practice. So there are 3 things that I concluded from my experiment. The first and second were that hydrocephalus adversely affects the dendritic morphology of pyramidal neurons, evidenced by decreased dendritic lengths and spine densities. The third was that the morphological changes I found can serve as a structural basis behind the deficits observed in this disorder, an understanding that can lead to more targeted and effective therapeutic interventions in the future. Thank you for your time and consideration.